Okay, we're going to move forward then to our oncology session. Let me introduce Dr. Mark Kirstein. Dr. Kirstein is an associate professor in the Department of Experimental and Clinical Pharmacology. His work revolves around kinetics, pharmacodynamics, and pharmacogenomics of anti-cancer drugs. And so Dr. Kirstein will moderate the session and introduce the speakers and take questions and answers. Dr. Kirstein.
Thanks, Dr. Jacobson. Good morning, everybody. So this will be the start of the first session on oncology, and so the way of thinking about this one is just a little bit different than the others in that most of the genetic alterations we'll be considering are of somatic nature rather than germline. There are a few exceptions to that. So our first speaker is Dr. Douglas Yee. He is a professor of pharmacology in the School of Medicine. He's also the director for the Masonic Cancer Center. He is having what I would say a very highly successful career. He started when chemotherapy was the primary treatment modality. And we have transitioned more to targeted therapies. And Dr. Yee has he has published over 90 papers just on focusing on the role of insulin-like growth factors signaling in tumorigenesis. And so these pathways that drive tumorigenesis are implicated as key targets in cancer therapy. And so what are the roles of mutations? And so this left Dr. Yee well poised to help develop this very novel clinical trial, the I-SPY2 trial, which I always enjoy hearing about. It's an adaptive platform design, and it's what I call a living, breathing type trial. 2 years ago, they published 2 key articles in the New England Journal of Medicine on neratinib and veliparib, and so you can refer to those articles. So I will present to you Dr. Doug Yee.
Thanks, Mark. There we go. Thank you for that very kind introduction. I do want to apologize in advance. I do have to leave after the talk. One of my pet peeves about conferences when the speaker comes, gives his talks and leaves, that's, kind of rude, and I never thought I would be that guy, but today is my clinic day, and for the clinicians in the room, there's no fury like the fury of your clinical boss when you cancel your clinic on short notice, so unfortunately I couldn't do that, and Shilpa knows that for sure. So I know that most of you don't think about breast cancer, and I wanted to spend a few minutes just giving you a background on what's happening in breast cancer, and I also know it's a little hard to see in this light, but these are mortality statistics for women from 1930 to roughly the present. This line is breast cancer, and you can see for many, many years, age-adjusted mortality was roughly similar to over a long period of time, but then roughly in the late 80s, early 90s, mortality started to decrease. And I know it's hard to see this, but breast cancer mortality is as low now as it has ever been. That's not because there's less breast cancer. If there's anything, there's more breast cancer. And you could ask the question, why is that? So this is a simulation from biostatistician Don Barry that he published several years ago. Don Barry's name will come up again in this talk, but what Dr. Barry did was he decided to model the trends if nothing happened, meaning there was no mammographic screening or no systemic adjuvant therapy. What he did was model screening only, adjuvant therapy only, and then both together, and found that screening and adjuvant therapy reduced the risk of breast cancer death. I am not going to talk about screening at all today. It's a relatively controversial area on how much screening contributes to the reduction of breast cancer mortality, but this is what the models suggest. So again, I know most of you don't think about breast cancer therapy, so I wanted to say a few words about adjuvant therapy. Adjuvant therapy is a term that was coined by the surgeons because for 100+ years, breast cancer was a surgical disease. It really took the majority of my first part of my career figuring out what they meant by adjuvant therapy. And what they meant is when you give medical therapy after the surgery, it makes their surgery better. So the surgeons coined the term and called it adjuvant therapy, but really it is medical therapy for breast cancer. And the principle is you want to eliminate or suppress micrometastatic disease in distant organs. So for many women who come to you, those micrometastatic sites are already established. And if you do nothing but the surgery, you will then have a relapse in years, months later, which is really the inattention to the metastatic systemic disease. We know Now we can exploit a couple of targets, estrogen receptor alpha and HER2 oncogene are excellent targets in breast cancer, and I think that that has been a model in my mind for targeted therapies, understanding estrogen receptor function. And I also think, and I'll try to emphasize, we need to individualize risk for each patient to provide appropriate therapy. So we can de-escalate therapy for low-risk patients, and we can provide precision therapy for those at high risk. My second apology is this is really not a pharmacogenomics talk because I know you guys are here for a pharmacogenomics event. For us, pharmacogenomics has had some insights. You heard a little bit, I think, in the previous talk about CYP2D6, so there's a story about CYP2D6 in tamoxifen metabolism and response, and I will just comment. There's a very large literature on this, and for the most part, we don't do this routinely when we prescribe patients to tamoxifen. We don't exome sequence very much either. We know that mutational tumor burden in breast cancer is low, and most of the breast cancer studies have not shown that sequencing and defining mutations has been particularly useful. We do know that a couple of germline mutations, BRCA1 and 2, probably indicate specific therapies. So we do at least do that, and particularly in high-risk patients. For us, the most important thing that's happened in terms of omics is gene expression profiling. And it's been used for estrogen receptor alpha positive tumors. So you know that most tumors express estrogen receptor alpha. It is also a hormone-dependent transcription factor. So if you said, I want to pick a genomic test that's going to tell me whether estrogen receptor alpha is activated or not, it would be transcriptional profiling. I've listed the multiple commercial products that all have a slightly different technology, but they're really all based on transcriptional profiling of estrogen receptor alpha breast cancers. So I want to spend a few minutes talking about the Oncotype DX. The Oncotype DX is also known as the 21 gene recurrence assay. This was probably the first test used, and the advance was that the company that made it figured out how to do RT-PCR on paraffin blocks, and that was a big advance because, as for the scientists in the room, you know the RNA you get out of paraffin is horrible, but the fact that it's uniformly horrible in the sense that it was not biased to degradation of any specific transcripts allowed Genomic Health to create this assay. So at the beginning of the month at ASCO, we heard the results of TAILOR-X. So TAILOR-X was a clinical trial which tried to ask the question, in women who are estrogen receptor alpha positive, do they really need chemotherapy? And can you use a genomic profiling test to help you make that decision? So here's the trial design. The trial design is women who had lymph node negative tumors, these are low-risk tumors that were estrogen receptor alpha positive, all received the Oncotype test. So roughly 11,000 patients were registered or enrolled, 10,000 patients were registered. Previous data that I'm not going to go into showed that the Oncotype score, which ranges from 0 to 100, if you were less than roughly 10, the benefit of chemotherapy for that subgroup would have been extraordinarily low, so those patients were not randomized. If you had a higher score, were 26 to 100, the benefits of endocrine therapy or tamoxifen or estrogen receptor blockers were not particularly useful too. So those women were also not randomized. They were assigned to receive chemotherapy with hormonal therapy. The dilemma was the women in the middle, roughly 6,700, so the majority, 2/3 of the patients fall into this clinical dilemma of whether they need chemotherapy or not. The experimental arm was endocrine therapy alone. The standard arm was endocrine therapy plus chemotherapy. The standard of practice up until this time is everybody got chemotherapy. One of the major groups that had done these trials showed that in lymph node negative estrogen receptor positive patients, women who got hormone therapy alone versus chemotherapy and hormone therapy, the chemotherapy and hormone therapy arm was always superior, and it was like a teeny weeny bit superior. So if you think about this entire group, there are obviously women who benefited and women who didn't, and that's what this study was trying to dissect. So these are the results. So remember, Arm A was the low-risk group who got hormone therapy alone. They did extraordinarily well, a 3% distant recurrence event, and that's what this is. So I said to you that breast cancer is lethal because of distant recurrence, so the primary endpoint for this trial was distant recurrence-free survival. In arms B and C, remember, these women were randomized to chemotherapy and hormone therapy or hormone therapy alone. There was absolutely no difference in the outcome. And in arm D, they saw that even those women got chemotherapy, they had a 13% distant recurrence rate. So this was important for us to do in oncology. One reason is because it was the first sort of predictor that we could back off on chemotherapy. And secondly, that was an important advance, backing off on chemotherapy. I said to you the standard practice prior to this was everybody got chemotherapy, and so now we're in an era we can think about de-escalating those for those patients that are not at risk. So what we learned so far was low-risk estrogen receptor alpha patients can be measured by genomic expression profiling, can be spared chemotherapy. There was an earlier trial called MINDAC using the MammaPrint assay. It's a 70-gene assay that showed basically the same thing. Low-risk patients have very little benefit from chemotherapy. But it raises the question, then, what's the optimal therapy for everybody else? And I just want to come back to this results slide. So this is the high-risk patients who got chemotherapy and hormone therapy. And I should have said in the beginning, the Oncotype DX was created to predict prognosis. It does not, strict prognostic factors did not predict benefit from any given therapy. So what Oncotype showed that it's true. If you have a high score, you have a poor prognosis. But not only do you have a poor prognosis, you have a poor prognosis if you get the best of the state-of-the-art therapy today. So I think most women would argue that a 13% distant recurrence rate despite state-of-the-art chemotherapy and endocrine therapy is unacceptable. So we have to do better in that arm. So that's where I-SPY 2 comes from. And the idea behind I-SPY 2 is that maybe we should give this systemic therapy before surgery. So every patient on the trial I just showed you had surgery, then they had their tumor removed, then it was measured for Oncotype, and then they got their thing. So I'm going to argue, let's do a different sequence. Let's do the chemotherapy first if a woman needs chemotherapy, and then use that as a model system. So the things we know about giving chemotherapy first, long-term outcomes are identical for pre or post-operative chemotherapy. So you need chemotherapy at some point. It doesn't matter when you give it. An advantage of giving it earlier is because some women who were destined to have mastectomy because their tumors were too large can be converted to a breast-conserving lumpectomy. That's an important advance. Clinically, I also think it's an important advance for women to give them some time to think about exactly what surgery they want. So the first decision is not surgery. The first decision is thinking about chemotherapy and then later thinking about surgery. The most interesting thing to me, and this would seem like a no-brainer, but you can identify tumor responses if you leave the primary breast cancer in place. And you can have pathologic complete responses, which means no evidence of tumor in the breast or lymph nodes after resection. So we call that PathCR, or pathologic complete response. And again, a no-brainer would seem to be that if you have a PathCR, you're going to do better than if you have a lot of residual tumor after you get your chemotherapy. So that it seems to be obvious, and there's a little controversy around that I hope to at least show you the reason we believe PathCR is a good outcome. So these are some of the older trials done by the National Surgical Breast and Bowel Project, NSABP. And it shows you all of these patients that gotten chemotherapy. There are different chemotherapy regimens. In the dotted lines are those women who had a pathologic complete response. And the red line, the yellow line are the women who did not have a pathologic complete response. It's showing that, yes, PathCR was a reasonable good biomarker for the outcome you want, and that's survival. It was good enough that 6 years ago now, the FDA put out a guidance that said, you know, we'll think about your drug in the neoadjuvant space if it increases PathCR rates. So this was a guideline for industry. They said that they were convinced that the data for pathologic complete response was robust enough that they would give the accelerated approval for a drug if it increased the pathologic complete response over a control arm. The first drug that was approved under that guideline was pertuzumab, an anti-HER2 antibody. Pertuzumab, or its trade name Perjeta, was approved in the neoadjuvant space before it received full approval in the adjuvant space, and I think that gave a lot of biomedical people interest in this trial design. So I'd like to talk to you about I-SPY 2. I-SPY 2 is run by my colleague Laura Esserman. Laura is a surgeon at UCSF and really is the sort of mother of I-SPY 2, if you will, and she spent many, many hours torturing this acronym to get the I-SPY 2, but it was really an idea that we wanted to do a neoadjuvant trial, and we wanted to be very biomarker rich. So I'll show you what that means. And I know it may be a little bit hard to see in this room. But basically, we screen women that have tumors larger than 2.5 centimeters. They get a research biopsy, shown here in the little microscope. They get a research MRI because one of our biomarkers is a response by MRI. This research biopsy is sent off for one of these genomic profiling tests, the MammaPrint. So women who are hormone receptor positive, MammaPrint low risk, just like the Taylor X people I showed you, do not continue on this trial. We decided very early that hormone receptor positive, low risk by a genomic test would likely not benefit. And fortunately, that was the right decision, as both MINDAC and Taylor X showed that result. So if a patient is then gets through the screening, they come back to an adaptive randomization, and I'll try to explain that a little bit. Our control now is standard chemotherapy, paclitaxel, followed by anthracyclines, adriamycin, and Cytoxan, and then there'll be multiple investigational agents in the trial. So everything I'll show you, or most of the things I'll show you today, were in combination with paclitaxel, and there can be up to 5 agents in the trial at any given time. There is another biopsy 3 weeks into the trial, another MRI at the end of 12 weeks, another MRI at the end, and more tissue if there's residual tissue at the end, and this is a pathologic complete response is our primary endpoint. I do want to comment that the adaptive randomization basically means that the randomization to any given arm is not fixed. Since pass CR is our readout, we can adjust the randomization engine, if you will, to favor those patients who had a pass CR. I will show you an example of this today. If you have a drug in a trial and you have it across all subtypes of breast cancer, and it only works for triple-negative breast cancer, then the randomization will modify that and assign the next patient who comes in with triple-negative breast cancer a higher likelihood of receiving that experimental agent. This is Don Barry's design. Don Barry is a Bayesian biostatistician, and I think the adaptive randomization gets you to an endpoint much quicker. So with that background of I-SPY 2, I wanted to show you the drugs that we've had in the trial. So roughly we opened 2010, going through the trial now. We have 11 agents in combination. I'm going to show you an analysis of the results so far. You will note that any given time, drugs can come and go. I'll explain to you when drugs graduate. So the light blue lines are drugs that have graduated or proven to be more effective at control. The kind of whatever tealish color here is drugs that did not progress, not graduate, were not better than control arm and drop as a futility, and they're drugs that are ongoing. So I want to just show you the reason I like I-SPY 2 is, one, every patient can participate. We're not targeting to a little narrow slice of subtypes. Secondly, we're very agnostic as to the mechanism of the drug. So I'll spend a little time talking about narantinib, but you know narantinib is an EGF receptor HER2 tyrosine kinase inhibitor. Veliparib is a PARP inhibitor. MK-2206 is an AKT inhibitor. SGN-LIV1A is an antibody drug conjugate. Pembrolizumab is a checkpoint inhibitor. So we've been very open to having multiple agents in the trial. My job actually on this trial is I'm the chair of the agent selection committee. I'm kind of responsible for trying to find new agents and making sure there's safety data that can be applied to this population. So one of the controversies has been, how good is pathologic complete response as a biomarker for a long-term outcome? And these are data we published at our breast cancer meeting, or we showed at a breast cancer meeting in San Antonio last December. So first off, these are the subtypes of breast cancer. This is hormone receptor negative, HER2 negative, hormone receptor positive, HER2 negative, HER2 positive, hormone receptor negative, HER2 positive, and hormone receptor positive. So first, I think everybody appreciates in any disease that breast cancer is not heterogeneous. There are multiple subtypes, and you have to think about multiple subtypes when you do a trial design. The other thing I'm showing you is the pathologic complete response rate throughout the trial. So it didn't matter what drug you got. Even if you got a drug that failed and you achieved a pathologic complete response, you're included in the statistics. So the reason I'm showing you this is to convince you that complete disappearance of tumor is a good idea. Now, it varies between subgroups. You see that the best acting subgroup is the HER2 positive subgroup that are hormone receptor negative. they have the highest rate of pass-CR. They're the smallest slice in our picture here, but it varies across subtypes. And the worst, if you will, is the hormone receptor positive, HER2 negative. So even though we selected these genomic high-risk patients within our trial, this is a subgroup of breast cancer that still needs some work. So showing you that together, I'm showing you now event-free survival and distant recurrence-free survival divided by pathologic complete response or not. And I would argue that a 94-95% 3-year event-free and distant recurrence-free survival for this genomically high-risk group of patients is a good outcome, and we are pushing to include pathologic complete response as the endpoint for our trial and as we move forward. You also see that it doesn't matter which subtype you have, even though hormone receptor positive, HER2 negative are least likely to have a pathologic complete response. If women have that, they do very well, and they all group together in 95-ish% range. In contrast, women who do not have a pathologic complete response, and most striking to me is the hormone receptor negative HER2 positive subgroup, have a very significant risk of recurrence over time, and that needs to be addressed as well. So what we've learned so far from this particular trial is that path-CR is highly correlated with the long-term outcomes we want, event-free survival, distant recurrence-free survival. And the value of path-CR, so again, even though the percentages of receiving a path-CR is not consistent, the value of PCR for event-free and distant recurrence-free survival is consistent. Pathologic complete response on any therapy, regardless of what you got, has equivalent correlation with event-free survival, and path-CR, we would argue, should be the endpoint for all patients. Therefore, and this is the therefore that takes a little bit of leap of faith, is that therapy should be changed for women unlikely to have a path-CR on assigned therapy. So in other words, the good news was path-CR is great. Bad news was a lot of patients don't get path-CR, and if you believe that the pathologic complete response is the endpoint, then you should switch what you're doing. So I do want to show you the nerantinib data. Nerantinib is a tyrosine kinase inhibitor. It inhibits EGF receptor and HER2. And we had it in all subgroups. And the reason we had it in all subgroups, because there's a reasonable story that the epidermal growth factor receptor has activity in triple-negative breast cancer, and it might work there. There's also the same story in hormone receptor positive breast cancer, and it might work there. And to me, the advantage of an adaptive trial is if that were wrong, you know, we have a hypothesis it's going to work on triple-negative breast cancer. If that were wrong, the trial will adapt away from those patients. In other words, fewer patients will be assigned. I mentioned to you, how do we determine when a drug graduates? So it is not simply the probability of adding nerantinib to chemotherapy is better. It is a probability of success in the phase 3 trial. So this is a very pragmatic endpoint that Don Barry put together. And it is if you do a standard randomized trial of 300 women assigned to the drug or not, you will reproduce these results. So that's where this trial came from. And if you notice, nerantinib, we set the threshold at 80%. Some late arrivals made it slightly less than 80%, but nerantinib only graduated in the HER2 positive hormone receptor negative trial. For those of you following the biomedical industry, you know that Puma went on to get this drug approved in post-adjuvant setting. That's a different story. But nonetheless, there was good activity in the hormone receptor HER2 positive subgroup. These are what the data look like. If you look at nerantinib in all patients, you find that there's a 33% pass-CR rate, 23%. So it did not graduate in the all patient subgroup. Here's the hormone receptor negative HER2 positive, where the predicted probability in the phase 3 trial was 79%, 56% estimated pass-CR versus 33% for control, and that's where the drug graduated, and that's how we published it. Now, again, I said we wanted to see nerantinib work in the triple-negative subgroup, and it was flat-out negative. It also was flat-out negative in the hormone receptor positive subgroup. So one disadvantage of our categories is they're still not finely enough parsed. Personally, I truly dislike the term triple-negative breast cancer because it just displays our ignorance that we don't know what's driving these tumors. So that's what we are. And triple-negative breast cancer is probably multiple things. So one of the good things about I-SPY is you have the biomarker to look at. So I'm going to show you a very complicated slide, You guys have these slides hopefully downloadable. If not, just email me. This paper should be published any second now, and I'd be happy to give you a preprint. But it gives you a chance to look at lots of stuff. It gives you a chance to look at EGF receptor totals, mRNA, Stathmin-1. For whatever reason, I don't know if there are any Stathmin-1 experts in the audience, but Stathmin-1 was a good positive predictor of response to nerantinib. What I wanted to show you, and again, I realize this is a pharmacogenomics seminar, but I wanted to use some proteomics and phosphoproteomics because, again, these are kinase inhibitors, so you want to look at the kinase activity. And I just want to show you phosphorylation sites on EGF receptor and HER2. These two correlated, I know it's hard to see in here, with past CR rates. And this was pulled apart in the triple negative subgroup, which are demonstrated here by the dark blue lines here. And it is a little hard to look at this level of resolution. But I'll show you it slightly differently. And that is, in red, patients who had a past CR that had phosphorylation on one site or the other site, and they seemed to cluster for the nerantinib arm, the patients with past CR, it's in the green, if you had phosphorylation on both sides. So that to me is a very pragmatic answer. If you had triple negative breast cancer and you're a phosphorylated EGF receptor and HER2, you should get a tyrosine kinase inhibitor against those two receptors. And that's what this shows. If you break this out, roughly 80% of the triple negative breast cancers had a past CR compared to the control arm. Now, of course, we did not randomize on that, so this is a post hoc analysis, and take it as you will, but it would argue a little bit that for a subgroup of women with triple negative breast cancer, they did get the right drug, but because we are not parsing it finely enough, we just couldn't detect it. So to show you where we're going next is we want to both escalate and de-escalate. So if a woman has a pathologic complete response rate after her first assigned therapy, she'll go straight to surgery. If a woman does not, then we will then reassign them to the best drug that their tumor needs. So the example I just showed you, let's suppose a woman got control therapy and it turns out she had phosphorylation of EGF receptor and HER2. And if she did not have a past CR, then maybe she could get the nerantinib arm. The other thing we're toying with is the idea that you can go directly into the assignment and then just stop whenever you have a past CR. And finally, we want to avoid chemotherapy and de-escalate, so the standard chemotherapy in our backbone is now at the end. So to conclude, I think I-SPY 2 is a biomarker-rich trial opportunity. We exclude low patients, and we had the potential to reassign patients on initial biomarker study. We hope that we can de-escalate therapy, we pick right the first time, and for patients without an exceptional response, we can match tumor to drug. We can also study resistance pathway by multiple biopsies, and I also didn't talk about precision imaging, but that's an important part of the trial as well. So my very last part of this, I want to show this is a national trial. These are all the sites that are open. As I mentioned, I chaired a working group, and we've been fortunate to have a lot of biopharma partners as well as biomarker partners that support this trial. So thanks for your attention, and there may be a little time for a question or two.
Thank you, Dr. Yee. Are there any questions from the audience?
I was just curious, PATH-CR is a very valid endpoint, but is PATH-PR a meaningful endpoint also? Just a partial response and not a complete response?
Yeah, so I didn't go into detail, and this is a term of art that I always have to explain to patients, but we have a scale where we judge how much tumor is left called residual cancer burden. That sounds horrible. But anyway, RCB scores goes from 0 to 3, and they're graded based on how big the tumor is, how much microscopic tumor is, there's a lymph node involvement. So it turns out RCB 0 and 1, and this is something we actually just presented at ASCO a couple weeks ago, are probably just as good. So microscopic residual tumor is probably just as good as complete disappearance.
So I just had another quick question. So for HR positive, HER2 negative who don't achieve PATH-CR, do you recommend they should not get any neoadjuvant, proceed to surgery directly? Because they're not going to achieve PATH-CRs no matter what.
Yeah, so that's a question that we're discussing. One of the things I didn't show you today is that of all the drugs we have that had in the trial, the only drug that actually benefited the hormone receptor positive subgroup was pembrolizumab. It tripled the pass CR rate from roughly 12% to 35%, and we're trying to dig out exactly what the predictive biomarkers for that are. And since we don't know, we need to reassign patients who would benefit from a checkpoint inhibitor if they didn't get it at time 0. So not quite going to surgery yet. We're still working on it.
Monica Akri, and I am wondering about the categorization that we accept right now for breast cancer. What about expression profiling? Have you collected data on this to try and determine the subgroups within our existing categorization and their response rates?
Yeah, no, so I agree with you completely. That's the next shoe to drop for the trials that have been reported thus far. So the 17-gene assay, MINDAC, reported their results, but they have not broken down the genes they measured to identify predictive benefit of chemotherapy or not. That has to happen, and I actually honestly think we will have to change the categorization of breast cancer. I showed you some antibody drug conjugates. So one of the ones in the trial we had is CL Genetics drug targeted against a molecule called LIV-1. So if that drug works, and it works in LIV-1 positive tumors, then you suddenly have a new category of breast cancer, LIV-1 positive tumors that benefit from a specific drug. But I definitely think that's the way we have to go, and we have to harness the data we have already. And I know people are doing that.
There are other questions? So, Dr. E, if there's a little bit of time left. So of all the agents that have graduated, which ones have actually progressed to go on to these so-called phase 3 trials, these 300-person trials, and has that really increased the efficiency of drug development?
So there are, as I mentioned, Narantinib, one of the other things about trials is always a moving target. So when we compared Narantinib Taxol, we compared it to Herceptin Taxol, which was then the standard of care. I mentioned to you that pertuzumab got the indication, so suddenly there was a new standard of care, and the company Puma opted to get their approval in the post-adjuvant setting. The other drug that was reported first was Veliparib, and it was a Veliparib carbotaxol. That was a drug that is owned by AbbVie. They did the confirmatory trial, and it reproduced our results exactly, but the thing they did was they actually gave a Taxol carbo alone arm, and it was the carbo, not the Veliparib. So, you know, in some ways, we want to be more efficient in our design, so we've never had a Taxol carbo arm. So, on one hand, I'm happy that they reproduced their results. I'm actually a little sad it wasn't the Veliparib. It was the other chemotherapeutic agent.
Okay, well, if there are no other questions, thank you, Dr. Yi.
Thanks. All right.